The gut lumen is composed of many molecules, like water, sodium, and glucose, and has a high concentration of sodium. The epithelial cell has water, glucose, and potassium, and a high concentration of glucose. A carrier protein, SGLT1, which is also known as a sodium glucose transport protein, acts like a co-transporter for glucose and sodium. This SGLT1 protein is what connects the epithelial cell to the gut lumen. The sodium-potassium pump connects the epithelial cell to the extracellular fluid, which has a high concentration of sodium. Since the gut lumen contains a high concentration of sodium, the sodium concentration gradient transports glucose from the gut lumen to the epithelial cell. Due to the fact that the gut lumen is a hypertonic solution, it is natural for sodium to move from an area of high concentration to an area of low concentration. So, the sodium moves from the gut lumen to the epithelial cell, moving down its concentration gradient, carrying a glucose along with it. This is known as facilitated diffusion because a transport protein is needed in order to transport glucose. Because glucose was transported to the epithelial cell, it now has a high concentration of solute. Therefore, water from the gut lumen follows the glucose, moving from an area of lower concentration to higher concentration through the use of aquaporins. The epithelial cell can only have a low concentration of sodium, so the sodium-potassium pump regulates the sodium present in this epithelial cell. The sodium-potassium pump does this by pumping 3 sodiums out of the epithelial cell and into the extracellular fluid and bringing in 2 potassium molecules into the cell. Since these ions are pumped against their gradient, it requires ATP. And this concludes my demonstration.